bacterial transformations, where you can transform, and in biology, transform means to put new DNA into something. And the easiest living thing to transform is a bacterial cell. They're cheap and they're fast replicators, and every time they replicate the protein you injected into it, and their ribosomes are used to produce that protein. For example, bacteria. This is a petri dish, and bacteria usually, when they grow, produce these off-white colonies. But if you do a transformation of them, you can insert a glow protein taken from a bioluminescent living thing, and within a few days, they'll colonize, and under ultraviolet light will produce this green color. It's pretty cool. They can also cause other living things to glow, like rabbits. They can inject a glow protein into rabbits, and within time, their ribosomes will make that glow protein, and they will glow in the dark as well. But you have to wear ultraviolet glasses. It's not like you're going to see bunny rabbits hopping around at night that glow in the dark. Okay, so reverse. Transformation means to add new DNA into something. Here's the process for bacterial transformations. It looks hard. It is not. Here is a human cell. Here is a bacterial cell. Here is your gene that you want to transcribe by the bacterial cell. So here is your recipe. So you take your recipe, in this case it's human growth hormone. I've seen it a lot with insulin to make being diabetic a little bit cheaper. So they take the human protein, they cut it out, and then they take the bacterial cell and they take the plasmid. You do not want to take this squiggly bacteria. This is called the nucleoid, and this has life-giving and life-necessary genes for the bacterial cell. If you cut it, the bacteria will die. You don't want it to die. You want it to reproduce. So you use the plasmid, which has extra unnecessary for life DNA. So you take your recipe, you take your plasmid, you cut them with the same molecular scissors. So if you can see, they fit together like puzzle pieces. You then cause them to come together, and through a series of lab steps, the bacteria absorbs the plasmid, and the ribosomes are used to mass produce that gene right there. And it could be converting the gene to human growth hormone or to insulin, etc. So bacterial transformations are like little assembly lines for human proteins. But my favorite analogy when I think of this is these yummy juice-filled beads at Yogurtland. Think of these beads as the bacteria and the juice inside as the protein it just produced. And once the transformation is done, the insulin or the growth hormone or whatever you cause a transformation of can be extracted from these bacterial cells, purified and bottled, and voila, you've got affordable medicine for many people who need it. So I'm sure there's more to that process with the purification than I said, but you get the gist of it. Harnessing bacteria to produce necessary human proteins.